In this video, we will discuss how to integrate Visium data with a single-cell RNA-seq reference dataset. First, we're going to subset the Visium data to make it more suitable for the integration step. The reference dataset we're going to be using was generated from the mouse cortex, so we will first subset the cortex spots in the Visium data. This way, the integration can give more reliable results when the reference is from the same tissue. We're going to subset the data with the tool v4 subset out anatomical regions based on clusters. In the parameters, you can choose the clusters to be subsetted. As a result, we will have the Seura object that you can give to the next tool. We will also have the PDF file containing the visualization of the remaining clusters on top of the tissue image. Moreover, before the integration, you should also make sure that the cell type annotations are found in the single-cell reference object. They should be located in the subclass column of the metadata slot. You can also check this using the tool Extract Information from Seura object in Gipster. If we now first choose the reference object and then from the parameters we can choose to print the metadata table. From the resulting table we can now look for the subclass column and see that the cell type annotations are found there. After these steps we can now finally integrate the spatial transcriptomics data with the single cell reference. Since Visium spots contain multiple cells, we can use the knowledge acquired with single cell analysis to explore the spatial data. With integration, we can predict the composition of cell types in the Visium spots. Moreover, with integration, we can locate the cell types in single-cell data in space. Here in this photo from 10x Genomics website, we can see how one Visium spot contains multiple cells, but with the annotated single-cell reference set, we can predict the underlying cell types in the spot. We're going to be using an anchor-based integration method by first normalizing the single-cell data and the subsetted Visium data with SC transform, and then by performing label transfer to transfer the annotations from the reference to the query set. The labels are transferred by first finding anchors between single-cell and spatial transcriptomics data with the mutual nearest neighbors. Then we use these anchors to transfer the information, aka the annotations, from the reference to the Visium data. These annotations are then visualized in a UMAP plot. You can run the tool v4 integration with single-cell data by giving the reference set and the Seura object from the previous step as input. For the reference, you will need a single-cell RNA-seq dataset that is similar in biology than the Visium data. As a result, you will get the integrated Seura object and the PDF file containing the plot of the annotations in a UMAP plot. You can also further visualize the results of the integration with the tool Seura v4 Visualize Integration. With this tool, you can visualize any individual features on top of the tissue image with spatial feature plots. Moreover, you can also identify and visualize cell types whose location is spatially restricted based on the computed cell type prediction scores. The spatially variable features are computed based on the marked point processes in a similar manner than in the step Identify spatially variable features, but with the exception that the cell type prediction scores are used as the marks rather than the gene expression. As a result, you get the PDF file containing the spatial feature plot with the chosen features. In the same file, you also have the computed top 4 cell types whose location is spatially restricted. Note that the results are only predictions based on the reference set and how well it covers the cell types that are present in the spatial data.